A very good afternoon, everyone. So today I will be presenting my journal on the topic Hominin fossils from dry and dry moron info, Paranthropology robust, craniofacial ontogeny. And this is an article which was published in the year 2023 in the journal named Science Advances, which has expanded Acro 14.1. So the terminology which we need to know before moving on to this is ontogeny is the growth and development of an organism, and hypotic is a representative fossil specimen of a taxon, and modularity, which is integration, it is nothing but independence versus coordinated growth anatomical regions, and synaptomorphism is shared derived traits indicating which always indicates a common ancestry. So the novelty of this study is it is the first detailed ontogeny series of P. robustus infants from these two species. So this journal it gives new insights. So it challenges previous hypothesis which has been put forth about cranial growth patterns and phylogenetic relationship between P. robustus, which is closer to Homo species than A. africanus. So there is this methodological innovation which is also seen in this JC, which combines the micro-CT, geometric morphometrics and logistic regression to assess the developmental changes of the specimens. So the aim of this study was to elucidate craniofacial growth patterns in P. robustus infants. The objective was to describe new fossils to fill gaps in ontogeny series, to compare the growth trajectories with A. africanus and early Homo species, test the hypothesis about modularity versus integration in cranial development, and to re-evaluate the taxonomy of disputed specimens in SK54. So, moving on to the materials and methodology. So, the primary samples were 4 Paranthropology robustus infants. So, there was one specimen from dry molen and 3 specimens from farm dry species. So the one named DMS47 was a fragmentary maxilla with deciduous teeth, and the one from named KW6420 is a mandible with developing teeth, and the other one is a best preserved maxilla with full deciduous dentition, and the other one is a bone with a cranial base, temporal bone, and occipital fragments. So these were the 4 samples which were taken. And the comparative fossils were from the Australopithecus species, that is a cow child, and two from the pan species. One is from the pan troglodytes, which is chimpanzees, another from the bonobos family, another from gorilla species, and another from modern humans. So these 4 was then compared to the other 4. So they have taken the species and then micro-CT scanning was done. They scanned it at 10 to 36 micrometer resolution. So they used for dental development staging with two-shot fusion analysis and trabecular bone density assessment was done. Developmental staging was also carried out in the study. Dental development was staged using the 14-stage scale, which was from Morris et al. Study, which was published in 1963. They assessed for crown initiation to completion pattern. So the molar emergence, which was used, it was used as a key developmental milestone in all the fossils which were achieved. Skeletal development patterns were also studied. The subarcuate fossa patency, that is the percentage of opening related to the semicircular canal length was studied, and logistic regression was predicted using the molar stage of molar development based on the subarcuate fossa opening in African species. So, and then morphometric and statistical analysis was done. Initially, they began with the geometric morphometric. So, 18 landmarks to landmarks were marked of HSCC, that is the horizontal semicircular canal and posterior canal fossa, and 79 landmarks or semi-landmarks for subnation morphology was marked, and between group comparison was done between all the species. The endocranial volume, EEV volume, was also estimated using 2 methods. One was the fossil-based scaling that compared the one species and gorillas with that of gorilla species. Regression model was also used which study the endocranial volume. So in statistical analysis, as I said previously, they used logistic regression for the molar development prediction. So the model used says that probability of molar beyond the root initiation was given in the Hominin. So for training data they used 20 African eggs with known molar eruption patterns. The results stated that there was a low probability of molar eruption beyond root initiation. So they also did principal component analysis PCA, and after which they estimated the EV estimation regression, which stated that the prediction was it overlapped the fossil scaling estimate. And moving on to the results, so these are the summary of the events, summary of the events which were seen in the study. Firstly, there is this maxilla growth. So P. robustus infants, they showed independent growth modules with early incisor suture fusion unlike that of Africanus species. Secondly, we have anterior pillars present before the molar eruption, which is supported by a dense trabecular bone, which indicates early masticatory adaptation of all the species. Palatal thickening was also noted, so that developed post molar emergence, which was contrast with Africanus species. So that was where in Africanus species near adult thickness was seen in specimen. Brain size was approximately 80% of its adult size during the molar emergence, which resembles that of current Homo species. Posterior cranial fossa was enlarged and inferiorly rotated like that of Homo species, suggesting accelerated occipital lobe growth. Dental development, according to it, deciduous permanent incisor proposition, that is 74%, it was matched to modern humans, unlike the tongue, which was only 48 to 60%. And then the P. robustus species, when reclassified with SK54, using the supraorbital traits, it aligned better with that of the Homo species. And then using the statistical support, logistic regression between the models and PCA analysis, it confirmed ontogenic distinction from species of Africanus. Moving on to discussion. So that is this craniofacial modularity. That is the premaxillary and maxillary regions, they grow independently, unlike previous singular modeling hypothesis. But palatal thickening, so anterior pillars appear early, but palatal thickening occurs later. There is brain growth patterns as said. It was 80% adult brain size by molar emergence, and there are some taxonomic implications which supported paratrophosis to Homo relationship than that of Africanus species. So there are also some controversies and debates existing. So one of it states whether does P. robustus share a recent common ancestor with the Boise? So we need more Boise taxa infant data to deal with that. And they also have they compared with previous studies also. So there are challenges in nasomaxillary singular modeling hypothesis, and anterior pillars as synapomorphies with that of Homo population. It is also compared with that of a study. The main strength of this study is it integrates paleontology, developmental biology, and phylogenetics. So it gives a holistic approach. So there is technical rigor where they use micro-CT and fragmentary fossils. The impact it resolves the debates about P. robustus growth and taxonomy. The limitation is that only 4 new specimens were studied, and the comparisons they only rely on single fossils. So there was a taxonomic bias which was seen. Fragmentation may obscure developmental signals, and there were assumptions also made in the study. So the EV estimates extrapolated from the limited comparative data. So the future direction could include including other samples like Boise and Ethiopian infants. So genomic insights can link developmental patterns to regulatory networks, and biomechanical modeling can be done to test the hypothesis about mastication. So these are the major conclusions. So the major conclusion is that premaxilla and pala, they grow independently. It was contraindicating with the prior models which was put forth. And the implication is that this study demonstrates microcity plus geometric morphometrics, which can be a gold standard for fragmentary fossil analysis. The bottom line is that Paleoanthropology's robust influence, they reveal a striking Homo-like, that is human-like growth pattern, challenging their link to Australopithecus and suggesting that they were an evolutionary side branch which is actually closer to humans. So STF prefers declassification as early Homo can further reshape the human family tree. So this is the checklist. So this study, it was adapted from the paleontological reporting standards from the SOAP or TRIO guidelines. So the title and the abstract introduction methods are all clearly specified, and then the fossil samples, it lists sites but it lacks taxonomic tamphotic damage details. The variables, data sources and bias control was all clear. Statistical analysis also well done with the logistic regression and scaling models. The results were, since there were fragmentary fossils, it limited the metrics, so the results were like a bit partial. The discussion and the finding details are all proper and it was all disclosed. Thank you.